Today we're going to discuss the second part of Chapter 4, where we talk about the organelles involved in protein trafficking. But first, let's review our lesson from the previous day. Can you name 3 differences between prokaryotic and eukaryotic cells? Go ahead and pause the video right now, see if you can answer this question, and then continue on. So were some of the main things that you named that prokaryotic cells do not have a nucleus, but eukaryotic cells do? Did you also mention that prokaryotic cells are fairly small and that eukaryotic cells are a lot larger compared to prokaryotic cells? And another distinguishing feature is that prokaryotic cells have ribosomes but no other membrane-bound organelles, while eukaryotic cells do have ribosomes and membrane-bound organelles. Okay, moving on. So the homework last night was the cell organelles handout. Hopefully you used your resources like your textbook and Google to be able to fill out this chart and learn some about the different organelles. If you have not yet completed this assignment, please pull that out and you'll want to follow along with this lecture and fill out this handout while we're discussing many of the organelles that are on the sheet. Okay, today we're going to discuss the endomembrane system, and you can see a diagram where all of these organelles involved are membrane bound, and it's endomembrane because they're membranes within the cell. So we're obviously talking about a eukaryotic cell since there's a nucleus and membrane bound organelles. A prokaryotic cell would not have any of the things showing in this picture. Today we're going to discuss how proteins are made, the trafficking or movement of these proteins through the cell, and how there are specialized organelles to perform metabolic functions. First of all, let's talk about the endoplasmic reticulum, also called the ER. It's a network of membranes and sacs, and there are 2 main types, mostly distinguished by their appearance on images seen with electron microscopy. The first type is called rough ER or rough endoplasmic reticulum. In the diagrams you can see that there are ribosomes studded on the surface, so the surface is not smooth, it's rough because of the ribosomes. And the main job of the rough ER, because it has those ribosomes docked on there, they'll involve lots of things with proteins. So the rough ER will package proteins, modify them, get them ready for secretion or export from the cell. And the vesicles will also bud off of the rough ER containing these proteins and send them to the Golgi. And also a replacement membrane is made at the rough ER. The other type of ER is the smooth ER. Clearly, based on the surface name, the surface is going to be smooth. There are no ribosomes on the surface, so a lot of the other functions involve the other macromolecules that don't involve proteins, so namely carbohydrates and lipids. So the function of the smooth ER is to synthesize these lipids, to metabolize carbohydrates that might be brought into the cell, as well as external environmental factors such as drugs and toxins and poisons. The smooth ER will contain enzymes to be able to break down and detoxify drugs and poisons. Another important feature of the smooth ER is to be able to store calcium. This is especially important in muscle cells for muscle contraction, and these smooth ER are actually called sarcoplasmic reticulum in muscle cells. Here's a diagram showing the rough ER and the smooth ER. So here's the nucleus, kind of in that purple. You can see the nuclear pores. And the outer membrane of the nuclear envelope is going to be continuous into the ER. So you can see it kind of folds right into there. So the parts of the ER that you see have those little dots, those ribosomes, that's the rough ER. Whereas the smooth ER, you're not going to see the ribosomes on there. The image on the right is actually from electron microscopy, and you can see the difference between smooth and rough ER in this image. So once proteins are made and packaged in the rough ER, they are going to bud off of the rough ER in little vehicles called vesicles, and these vesicles will dock with the Golgi. So the function of the Golgi is to synthesize and package materials, small molecules for transport in vesicles, and to produce lysosomes. So the Golgi contains a series of flattened membrane discs, and the inside of these discs is called the cisternae. And as vesicles approach the Golgi, they will merge with the side of the Golgi that actually faces the nucleus. So that's called the cis face. It receives the vesicles. And as materials inside the Golgi are packaged and are going to leave the Golgi, they will bud off within vesicles again through the trans face. So that's the outgoing part of the Golgi. You can see this teal colored organelle right here is the Golgi. They're flat, and you can see the vesicles, which look like round spaceships with proteins inside of them. They'll dock with the cis face of the Golgi and then continue on and be processed within the cisternae or the internal parts of the Golgi. And then again, these vesicles will bud off and leave the Golgi. So you can see on the right side transmission electron microscopy of the Golgi apparatus. Now, one of the other functions of the Golgi or the rough ER is to be able to produce the proteins that will eventually go into lysosomes. So those proteins are mostly going to be enzymes, and they're going to be special enzymes that will break things down. So they're hydrolytic enzymes. So you can think of the lysosomes kind of like the recycling, cleaning organelle of the cell, and it will do intracellular digestion and recycle worn out organelles and materials, other proteins, so that they can be broken down to their most basic monomers and simple components and can be reused. Now, lysosomes also contain these hydrolytic enzymes, so if the cell were not to be doing well, or the cell were programmed to die and commit cell suicide or apoptosis, the lysosome would actually burst open and release those hydrolytic enzymes to be able to digest the other organelles within the cell. So the hydrolytic enzymes will be conducting hydrolysis, which is the breakdown of macromolecules. You can see here in these images, microscope images of lysosomes, and some of them on the right side picture right here that I'm pointing to is that you will see some damaged organelles that are located within that have been engulfed by the lysosome or fused with the lysosome. So you see part of a peroxisome and part of a mitochondrion, and those are worn out cell parts that are being broken down and recycled by the lysosome. Lysosomes also help with digestion for cells that are unicellular, for example, paramecia. And so these single-celled organisms can ingest food particles by the process of phagocytosis, as you see in diagram A here. And these food vacuoles will then merge with or fuse with the lysosome, and those enzymes will break down the food. You can also see here on the right side, diagram B, autophagy is where the cell is actually going to digest its own organelles. All right, review question number 2. Let's put into practice what you've just learned about lysosomes. What do you think would happen to a cell if it did not have fully functional lysosomes? Pause right here, think about your answer, and then continue when you're ready. So in the earlier slide, we talked about how lysosomes will digest worn out cell parts and other macromolecules and be involved in programmed cell death. So any one of those things might not be able to happen. There are cases of where there are individuals with a genetic disorder with non-functioning enzymes within the lysosomes, and what can happen is there's a buildup of these materials that should be broken down, and that can lead to clogging and congestion of a lot of extra materials in the cell and potentially lead to cell death when it was unintended, or cells might not be able to conduct apoptosis and commit cell suicide without those lysosomes. Okay, the next organelle we're going to discuss are called vacuoles, and the main purpose of them is to store materials like food, water, minerals, pigments. Like in plants, they may have vacuoles that contain pigments, and some cells might house poisons to separate the poisons from the rest of the cell so that it doesn't harm the cell itself. These are also membrane-bound vesicles, and you can have special vacuoles such as food vacuoles or contractile vacuoles. Those might be found in paramecia or single-celled organisms. And then plants have one large central vacuole, and you can see that in the image below. And the main thing that is located in the plant's central vacuole is water and some ions in there as well. And the main purpose is to fill up that plant cell, to put some pressure on the outside cell wall of those plant cells to maintain turgor pressure. So this is what allows plants to stand upright, why they're not looking wilted. Celery is nice and crispy. It's because of the water that fills that central vacuole, providing turgor pressure. All right, so to summarize the endomembrane system, you have the nuclear envelope right here, where the outer membrane is continuous with the rough ER. Proteins are produced there and modified there in the rough ER. They can bud off of the rough ER through these vesicles, merge with the cis face of the Golgi, and continue the packaging and processing for these proteins to either stay in the cell within organelles like the lysosomes, or they can be shipped out and exported or expelled from the cell. Once leaving the Golgi, these products will leave in a vesicle that buds off, and if these vesicles stay within the cell, they could turn into organelles such as lysosomes, or these vesicles could be shipped to the cell membrane or plasma membrane, fuse with the plasma membrane, and then you expel the contents from those vesicles outside of the cell. So examples would be the hormone insulin, which is produced by the beta cells of the pancreas, and those cells would be producing a lot of insulin hormone and then be excreted from those cells. All right, next we're going to talk about some specialized organelles found in plant and animal cells, or one or the other. And so I just wanted to point out that this is an animal cell, and you can see some of the really common features that we talked about before, but you're also going to see an area called a centrosome right here that is found in both plant and animal cells, but you're going to see there's these specialized structures called centrioles. Now those structures are only found in animal cells and not plant cells. We will talk about later how the centrosome will have microtubules, or those spindle fibers will originate from there for cell division later on. Some other unique things that you will find in animal cells are these lysosomes as well, and you will see the mitochondria, which are in both plant and animal. You'll see cell membrane here, but no cell wall. Some of these animal cells might have these microvilli, for example, such as the intestinal epithelial cells, and some animal cells might also have be able to move or swim with the presence of flagella. Notice this list on the right, bottom right, where it says not in animal cells. You'll need to be able to know the differences between plant and animal cells later. Okay, now this is a plant cell. Some distinguishing features. You will see a really enormous large central vacuole here. And then not only is there a cell membrane, but there is the strong cell wall on the outside, as well as the presence of these green colored organelles, which are the chloroplasts. So mitochondrion and chloroplasts are really unique organelles. And let's talk a little bit about history, in that we think these organelles, or scientists believe and have evidence to support, that these organelles aren't just little membrane-bound organelles within a cell, but they actually were their own organism. They were prokaryotes that lived and survived on their own until they were engulfed by another prokaryotic cell. So mitochondria and chloroplasts share similar origins, that they originally were prokaryotic cells that lived on their own, and then they were engulfed by the ancestors of what we know today as eukaryotic cells. So to allow for cell specialization, for these cells to get larger and to become more efficient in what it does within the cell. So we want to present 3 key pieces of evidence to support the endosymbiont theory that chloroplasts and mitochondria actually were their own cells. First of all is that both mitochondria and chloroplasts, those are the plural versions, both have double membrane structures. So if you think about a prokaryote, a bacteria, that it has a single layer of the cell membrane. And if you were to be engulfed, you would have 2 membranes. The inner membrane would be the original cell membrane, and the outer membrane would be the host cell membrane. And so that's what you can see here, what would happen, how these organelles would end up getting 2 membranes. The other thing is when you look inside chloroplasts and mitochondria, they each have their own ribosomes and DNA. And it's DNA genes that are not found in the nucleus, and these ribosomes are made of slightly different material than the ribosomes in the cytoplasm. So these ribosomes allow specialized proteins to be made within mitochondria and chloroplasts, and the DNA has a set of genes to be able to direct the production of these particular proteins. Now, these proteins found in mitochondria and chloroplasts, not all of them originate within those organelles. Some of the proteins actually are coded for by the nuclear genes, and then those proteins are somehow brought into those organelles. Now, the last piece of evidence is that unlike the other organelles, which have to be produced within the cell and are single membrane bound and bud off of structures like the Golgi, mitochondria and chloroplasts actually reproduce independently within the cell. That is, they make copies of themselves in a fashion very similar to other prokaryotes like bacteria. They will divide by binary fission. They will make copies of their own DNA within there. It's a singular chromosome DNA, and then will bud off and separate to become 2 new chloroplasts or 2 new mitochondria. So they reproduce independently within a cell. And you can imagine that when they make copies of the DNA, there can be mistakes that happen, and so you can have mutations in different mitochondria that are not the same. All right, so let's talk about what these mitochondria are made of and what their purpose is for. Mitochondria, now please don't say the powerhouse of the cell. In AP Biology, we're going to now be defining mitochondria as the site of cellular respiration. Mitochondria have 2 membranes, so the outer and inner membrane. And the inner membrane is highly folded to increase that surface area. So you can imagine there's some sort of activity that's happening on that inner membrane of the mitochondria that we will find out later about in a future chapter. So all those folds are called cristae, and there are enzymes that are embedded inside that, within that inner membrane of the mitochondria. Within the mitochondria, there's fluid, and that is called the matrix. And so you can see the diagrams here of electron microscopy of the mitochondria, and then also a model or diagram picture of it. You can see that within it there's a circular chromosome DNA and ribosomes. So again, those are 2 pieces of evidence for the endosymbiont theory right there. Chloroplasts are also endosymbionts, and they are the site of photosynthesis. So being able to capture sunlight energy and convert it to stored chemical energy. They also have two membranes, and within the chloroplasts there are stacks of discs, and these discs are called thylakoids, and the stack of them is called a grana, or granum for one, and the fluid surrounding that is stroma. So we will go more into detail later when we discuss photosynthesis, but the reason that chloroplasts are green is that they do contain pigments of many different colors, but the predominant one is the pigments are chlorophylls, and they do reflect the color green, and they're really important for being able to capture sunlight energy. So here's the drawing of a chloroplast on the left, and you can see again there is circular DNA chromosome there, ribosomes of its own, a double membrane structure. And so just like the inner membrane of the mitochondria, the thylakoid discs have the thylakoid membrane to increase the surface area for photosynthesis. So again, structure dictates function. So increased surface area is a feature of the structure to be able to allow it to do what it needs to do. So you can see here in this electron micrograph of a diagram of a chloroplast right there. Okay, last review question. Which organelle, mitochondria or chloroplasts, are found in both plant and animal cell? And then what is only found in plant cells? So the answer here is mitochondria are found in both plant and animal cells, because all cells on Earth have to be able to do cellular respiration and to get energy in the form of ATP to use for cell processes. But in plant cells, we only find chloroplasts. So as cool as it might be to be green and to be able to do photosynthesis, we can't. Animal cells don't have chloroplasts. So that's a specialized feature of plant cells, which allows them to be autotrophs and make their own food.